This is our presentation on the Ebola virus by Ariel Vicara, Nicole Selinsky, and Marissa Kovalevsky.
What is Ebola virus disease, also known as EVD? It is a deadly disease with occasional outbreaks on the continent of Africa. It affects humans and non-human primates such as monkeys, gorillas, or chimpanzees, and it is an animal-borne pathogen that is believed to have originated in monkeys or bats. This is known as zoonotic transmission when a virus jumps from one species to another. It is caused by a virus in the genus Ebola virus, which has 6 viruses in total, but only 4 that cause human disease. These are Zaire, Sudan, Thai forest, and Bundibugyo. Zaire is the most dominant and fatal in humans, and EVD is a member of the Filoviridae family.
This is the basic Ebola virus diagram. The lipid membrane holds the virus particle together and is made of proteins and lipids from the human cell. The glycoprotein uses this to attach to the surface of human cells, and the polymerase uses this protein to synthesize RNA through replication and transcription. The viral RNA is the genetic material, and Ebola has 7 genes. The nucleoprotein is nucleic acid bonded to a protein. The virion structural protein makes up the capsid, and the glycoprotein induces endothelial cell disruption and cytotoxicity in blood vessels. There's also the RDRP polymerase, and the predicted order of the genes is 3' nucleoprotein VP35, VP40, glycoprotein VP30, VP24, and the RDRP polymerase.
Transmission of Ebola virus disease. It can spread through direct contact with blood, bodily fluids, or tissues of an infected or dead animal, direct contact with contaminated objects such as clothing, bandages, or needles, and it enters through broken skin or mucous membranes in the eyes, nose, or mouth. EVD can survive in bodily fluids for days at room temperature, and it can also survive on dry surfaces for several hours. EVD has an incubation period of 2 to 21 days, which is the time between infection and symptom onset, and a person infected with Ebola is not contagious until onset of symptoms. Because of these ways of transmission, healthcare workers or family members and friends of infected individuals are at highest risk, and it poses little threat to travelers and general public.
So this is the infection of the Ebola virus in the human cells. At point A, the Ebola virus is enclosed in a package that contains RNA. B is the glycoprotein of the virus that sticks out of the membrane. It's seen in green, and it binds to the receptors in red on the cell surface. At C, the binding of these receptors triggers a cell-eating process called macropinocytosis, which results in the virus being engulfed by this wave-like motion of the cell membrane. And at point D, once it's inside of the cell, the virus RNA is uncoated, at which point it hijacks the human cell's proteins to create more copies of itself. At E, once new viral particles have been assembled, they move into the cell membrane and bud off, at which point they can travel and infect new cells, as seen at point F.
This diagram shows some common symptoms of Ebola, some including gastrointestinal symptoms like abdominal pain and vomiting. Also, unexplained hemorrhaging can occur, and bleeding and bruising. In fatal cases, patients will experience early onset of severe symptoms with progression to multi-organ failure and septic shock. In non-fatal cases, patients typically start to improve between 6 to 11 days after onset of symptoms. The virus can show up in blood or semen of a recovered individual for up to 10 years.
Some factors that impact survival rates are early identification, access to healthcare supplies and facilities, drugs against the viruses, new vaccines, and prior infection has shown to provide some immunity in several trials. You can treat symptoms by getting fluids in the body to rehydrate. You could get blood transfusions, proper nutrition, and breathing assistance as needed. In undeveloped countries, it's harder to have access to these healthcare supplies, so it is more of a threat.
There are currently several FDA-approved drug treatments to treat Ebola virus. The first approved drug was Inmazeb, which targets the glycoprotein on the surface of the Ebola virus. It contains 3 antibodies that can bind to the glycoprotein and block attachment and entry of the virus into the host cell. Another treatment is Ebanga, which is a human monoclonal antibody, which is a genetically engineered protein that acts like a natural antibody to stop infection and replication of the viral genome. Just like Inmazeb, it binds to the glycoprotein to prevent the virus from entering the host cell. In a clinical trial, patients who received Ebola had a 14.3% decrease in fatality compared to those who received a control drug.
Another potential drug treatment is galidesivir, which is an adenosine nucleoside analog antiviral therapeutic. It's effective against many RNA pathogens and filoviruses such as Ebola and other infectious agents such as MERS-CoV and SARS-CoV. The goal is to inhibit RNA chain growth and interrupt viral RNA synthesis. If the viral RNA polymerase incorporates galidesivir's nucleotide into the RNA chain, there will be premature termination of the viral genome chain and the virus can no longer propagate in the body.
There are vaccines being developed to prevent the Ebola virus, but they're only clinical trials and no true vaccine that is FDA approved. There's multiple vaccines that have been tested, as seen on this slide.
The first vaccine that was developed is genetically engineered vesicular stomatitis virus, which is an animal virus primarily found in cattle, and VSV is used to express the EBOV glycoprotein. VSV causes no symptoms in humans. It has a small genome and it's genetically manipulatable, which makes it a good expression vector for foreign particles such as Ebola. Phase 1, 2, and 3 clinical trials have been conducted in 3 different countries, and they have shown a safe, robust immune response with few side effects. The data also supports the use of this vaccine in emergency situations. The CDC, the World Health Organization, and the National Institute of Allergy and Infectious Disease have conducted and are still conducting research on this vaccine.
Another vaccine that was developed is the chimpanzee adenovirus vector to deliver the Ebola genetic material, and it was designed to protect against the Ebola Zaire species. Numerous phase 1 clinical trials proved to be safe and immunogenic, meaning that it produced an immune response in the body. Phase 2 clinical trial conducted in Liberia outbreak in 2014, and the Sabin Vaccine Institute has licensed CAD3 platform to develop vaccines against the Sudan and Marburg Ebola virus strains.
This next set of vaccines uses a heterologous 2-dose strategy, which is given 4 to 8 weeks apart. The first dose, AD26 vector-based vaccine, expresses a Zaire Ebola virus glycoprotein. Then the MVA-BN phyllo acts as a recombinant DNA molecule, which encodes glycoproteins and nucleoproteins from various Ebola strains. Phase 1 clinical trials were conducted in 5 different countries and concluded safe and strong cellular immune responses. A randomized and placebo-controlled Phase 2 trial was conducted in France, which again concluded that the antibody responses produced in nearly all participants increased over time. The anti-glycoprotein antibodies persisted for 1 year in 98 to 100% of all participants, depending on when the second dose was administered.
The basic reproduction number, or R naught, signifies the number of people that an infected person could potentially infect. R naught and lethality have an inverse relationship, meaning that the lower the R naught, the more lethal the virus is. Ebola Zaire has a relatively low R naught. It has a 1.51 to 2.53, and this is because it's difficult to transmit because you need to have direct contact. And it's more lethal, so you'll have less chance of infecting others, and asymptomatic people are not contagious. Compared to the Omicron variant of SARS-CoV-2, it has a high R0 value of 7.0 or greater, and this is because it's easy to transmit through your respiratory particles. It's not as lethal to the general population, but with increased age and comorbidities, it becomes more contagious. Asymptomatic people are also contagious, so they can walk in the general public and spread the virus.
This graph shows Ebola compared to other viruses in terms of contagiousness, which is on the x-axis, and lethality, which is on the y-axis. Ebola is not very contagious, but has a 30% survival rate, which is only slightly lower than untreated HIV or untreated rabies patients. The Omicron coronavirus variant to compare lies somewhere near the yellow dot on the graph, with a reproduction number around 7 and lethality of roughly 1.2%.
Thank you for listening to our presentation. We didn't get all this information from our own brain, so here are our sources.